First, I want to thank Larry for inviting me, even though he's known me for a long time. He still did. The laboratory, all the people who did all the work. So obviously, to us at the Broad Institute of Harvard and MIT, or the Broad Institute of MIT and Harvard, depending on where in Cambridge you are at the moment, is a critical issue for us because we're not sufficiently happy with just discovering this stuff. We want it to make a difference ultimately. And so that's why I want to talk about it a little bit. So some of this is going to be kind of recapitulating some of the stuff we did this morning. Some of it's going to be my own kind of unique spin on it. I actually have a script to follow, so I don't go too far off script because it's being televised. What I would like to do is welcome questions. I realize that some of the talks this morning were fairly science heavy, and it's my job predominantly to translate the science into language normal people can access. So if there's questions you have, this will be a good opportunity to do that. And I'll try to keep my remarks short enough that we can get some questions in. Okay. It works. So since the beginning of recorded time, we've been fascinated by heredity and realized that it was somehow tied to sexual reproduction. No, there are many theories about just what was being transmitted floating about. Although you really couldn't see what the elements of heredity were, you could certainly see its results in terms of traits being passed on, or what we would call today phenotypes. And they seem to run in families, just underlining the suggestion that there was something biological going on. Sometimes heredity is really obvious, and sometimes hugely productive, as we can see here. Sometimes heredity seems to have disappeared, resulting in an offspring outlier who doesn't quite fit in. I know that's not a real family, that's all right. And it's hard to imagine either Herman or Lily having an affair. It's also noticed early on that it wasn't just traits that you see, but the diseases tended to travel in families. This is the Tsar Nicholas II's son Alexis was obviously affected by hemophilia, struck all the members of the royal many members of the royal family going back to Queen Victoria. So it's really only in the last century, which is a fairly amazing thing to think about, in 1942, when we realized, when it was definitively shown that the stuff of heredity was DNA. This mysterious material turned out to be a rather boring molecule. About 10 years later, Watson and Crick pulled together a bunch of data from different labs, not their own, but cleverly put it together to define the structure of DNA. And DNA, as it turns out, is a fairly simple molecule as biological molecules go. At the same time, people began to realize that, gee, maybe finding changes in this hereditary material and this very simple structure could possibly explain or correlate with these phenotypes and explain diseases and traits. And this notion was buttressed just a couple of decades later when it became possible to identify specific DNA changes in single genes that could have devastating consequences, like Tay-Sachs or cystic fibrosis, the kind of low-hanging fruit that they talked about this morning. So this led, I'm going to have a slide that someone else used here. This led to this fairly simplified view of what genetics was, that the relationship of genotype and phenotype was somehow directly correlated. It's a stupid idea, but at the time, that's kind of what people were thinking. But we know now, and people even back then who were working on complex traits in animals like fruit flies recognize that it's not this simple. Although it'd be great if genotype correlated directly to phenotype, we could all go home and be well. It simply isn't the case, except for, like I said, a few of the rare mutations. I'll show an example of that in a bit. Instead, genotype is related to phenotype through this thing I call a black box, ultimately. The best description of it right now, because I can't think of a better word, is biological complexity. It includes all the environmental changes, it includes all the stuff that happens in biology once the recipe is read, it includes all the epigenetic changes, and epigenetics simply means literally on top of genetics. What are the things that decide what parts of a sequence are read or not read, et cetera. It all changes that play the role in how that basic recipe is translated into some phenotype. So if, the example I always use is if you bake a cake in Boston, where I live, versus a cake in Boulder, and you follow the same recipe, the cake's going to be very different. There's a lot of stuff that goes in, even though the recipe is the same. So within this black box hides the processes and interactions that lead to extreme and subtle phenotypic changes, as well as the causes of the various common diseases and the rare diseases. It also holds within it, why we're here, the potential for accurate diagnoses, for efficacious therapeutics, and even prevention. So the box is where the action is. So neither phenotype nor genotype are going to be the thing of the greatest interest, ultimately, in terms of understanding biology and medicine. But they're not invaluable, it's just they're not the whole story. Both offer, though, distinct ways into the box. The question, to my mind, as I think about this and for the purpose of this conference, is which of those approaches, genotype or phenotype, is actually medically more relevant, at least in the foreseeable future, at least in the future that many of us in the room still have. So I'd like to spend a little bit of time looking at both ways in, which we've done a bit over this morning, and trying to understand what their distinct contributions to this thing called personalized medicine might be. So let's start with genotype. So this is the Queen Victoria pedigree. You can see Tsar Alexis down here. Started way up at the top there. So why has there been such a tremendous focus here? Some of it's historical. First, it gets to the heart of our fascination with heredity. It is the stuff of heredity, as already mentioned. Second, it's been kind of a sexier field of study. Techniques and technologies developed really rapidly. But even more important are the scientific reasons why DNA has leapt so far out in front. So as I mentioned, DNA, that's not a live caption of Watson and Crick. That's just my heading on there. So I mentioned it's a very boring but a very long molecule. It's very simple in its structure and chemical makeup. It makes it very tractable to experimentation and manipulation. It's also really tough. So one of the reasons we can extract DNA from 80,000-year-old Neanderthal bones and see who they slept with is because the DNA is still relatively intact in places. In fact, it's funny, back when Avery and his colleagues defined DNA as the hereditary material, no one wanted to believe them because it just seemed too simple. It just didn't seem to have anything to match the complexity of biology, where most people believe proteins were, in fact, the element of heredity. So I'm going to skip ahead 50 years, and all that excitement of genetics that built up in DNA, ultimately, I'm skipping a lot of stuff like sequencing and things that happen, but all that excitement built up into the Human Genome Project. The development of two things, the technologies in the lab, but also the computational power, allowed people to really begin to think that we could take on the human genome. So the original Human Genome Project, when you think about it, it started 1990 or so. It took about 10 years to get to the first draft and about $3 billion, depending on whose website you read. And there was a lot of intrigue and a lot of very interesting characters involved as well, one of whom I work for, but we won't go there. It resulted in having a complete human genome, or kind of a mix of a complete human genome. All 3.2 billion bases, nearly all 3.2 billion bases of the A's and the C's and the T's and the G's, which are laid out on a computer for anyone to look at right now. So the draft sequence was published in 2001. The final, quote, final sequence was in 2003. And though it's not incredibly useful in its own right, it's an amazing accomplishment. And it laid the groundwork for this explosion of technologies that we've witnessed over the last 10 years, and even a bit of data, and especially a bit of hype. So let me talk about the whoops, did I miss my GWAS? There he is. So I wish Dick was still here, because Richard was still here. I was going to argue with him a little bit about some of this. So I want to explain GWAS in a way that's accessible to normal people. So sequencing, when it costs $3 billion to do a genome sequence, you're not going to do a whole genome sequence. But about the same time, a technology had been developed where you could. It was clear that what we really wanted to understand was not what the sequence is, but what are all the variations in all the different people and which ones go with which diseases and traits. And there was actually technology in place to look at those individual A and C and T and G differences, the things called SNPs. And so the notion began, let's catalog all those. And so this effort started taking place with the drug companies, and there's millions, 11, 15 million SNPs now that have been identified that are common ones in the various populations. And you could use these SNPs as kind of signposts across the human genome. The other thing that became really useful, so you wouldn't have to look at 11 or 15 million SNPs, is that because we haven't been around very long as a species, there has not been a lot of shuffling of the chromosomes, as was pointed out this morning. One SNP could be a marker for a whole bunch of SNPs on a single block, which is called a haplotype. And so instead of having to look all the way across at every one of the SNPs, you could look for that one SNP to identify with that whole block. The best analogy I can think of is Google Maps. You know there's a house on fire somewhere. And Google Maps maybe could get you to the block. And that's the whole point of the SNP thing. You could identify the block where the house was on fire, so you only had to look at whatever, 100 houses rather than across all the houses. So whenever they say genome-wide, you looked at SNPs across the whole genome. Association meant you found a block that was associated with a fire in there somewhere. And study was because you studied it. So GWAS is pretty straightforward. It was the workhorse because it was cheaper to do than sequencing at the time. But ultimately, we really want to look at everything. So let me talk about sequencing. I already went ahead here. So the challenge of sequencing has always been the cost, but that's entirely changed at this point. We're making it more, we're putting out more data, doing it faster, doing it cheaper. At the Broad Institute currently, we can do a whole human genome in about 2 weeks for about $15,000, as opposed to 10 years and $3 billion. And it's continuing to drop. So this is kind of how much a gigabase is, you know, a bunch of bases. How much production there's been year by year. And there's this amazing rise. So if you go back, so this point here is now this point here. So from 2006 to 2008, there was a tremendous jump. And then from that point in 2008 into 2009, there's that. And then this year, we're actually going to be doing that much. So this is all sequencing that's starting to come down the pipe. And you know, there's computational nightmares associated with all this. But it's all data, and it's all coming out of various patients with different types of diseases, et cetera, et cetera. At the same time, the cost of sequencing has dropped down to a ridiculously low amount because of the technologies that have come along the way. And those are some of the big technologies. There's an Illumina person here who should be very proud of seeing that as the Broad's main focus. In fact, we're at the point now where it may even be today, it's sometime this year, that the cost of sequencing a base pair is more cheap than the cost of storing it on a computer, which is just phenomenal. Crazy thing. So all this stuff, this amazing focus and obsession with DNA and sequencing and stuff has led to some really interesting social phenomena that we still don't have any idea what most of these data actually mean. So to be fair, there's been some clinical applications that appeared we've heard about. Things like being able to tell what type of cancer something is, maybe. To being able to dose warfarin, for instance, or Coumadin based on how you metabolize it. So there's been some input already from some of this sequencing. And these early efforts have led to kind of this explosion of interest in how DNA is going to redefine medicine or is redefining personalized medicine or making up a new thing about personalized medicine. And so I've got the PMC, which Nick mentioned, or Steve mentioned, who are really big on DNA is going to be the be-all and end-all. And of course, we know that's not true, but you can read their case study. The other thing that was interesting that came up briefly was the direct-to-consumer groups. And I managed to get Pathway Genomics up here because they're the ones who were going to make all their money at Walgreens. This whole thing is, it's fascinating to look at these sites because they claim in the fine print not to be offering medical advice, but everything else, all the marketing is about taking charge of your health. And the thing that's really obnoxious about this is that empowering people with data, which they say is what's important, is fine, but the data are not actionable. They're not well understood even by the experts, and they're only marginally actionable insofar as they're no better than what your grandmother told you to do. You know, eat better, get sleep, get exercise. I don't think you need to pay a DTC company for that. And the other dark secret about all this stuff is that it's ultimately only going to be able to provide a prediction of some kind of relative risk. Even as if we get it down to a complete art form, only relative risk. There's so much more beyond the recipe that has to be taken into account. So let me give you a couple examples of things that have been recent that actually kind of caught us up a little bit short. Hereditary hemochromatosis is kind of a fairly nasty disease caused by the body's overzealous uptake of iron, which is then stored in a lot of places like joints, et cetera, where it shouldn't be. Although it's not a disease a lot of people have heard of, it's not that uncommon. And in a kind of a nod to our medical past, the most effective treatment is drawing blood, bloodletting. So in 1996, a group of scientists in those early heady days of genetics and disease found that a mutation in a gene known as HFE, or high iron Fe, HIF, sorry, was responsible for the vast majority of the cases of this disease in the cases they studied. So it was presumed, based on that, you've studied all these people with the disease, and they all seem to have this particular mutation, that must be the cause of this disease. So they argued that this particular mutation had a very high penetrance. In other words, whenever that mutation appeared, the person would develop this disease. It's only recently, with the ability to scale up and do many more samples, that we've discovered, and was published in New England Journal recently, that in fact, the fact that they were looking at patients with the disease kind of helped them. When they began to look across populations of people that didn't have the disease or had very mild forms, there were lots of people, many in this room, 1 out of every 200 of you are carrying both of the bad copies of this gene for this disease, even though you may have no phenotype. So that's a bit of a taken aback shock. Even more so, the people that were carrying both copies of the disease only went all the way to disease about 30% of the time. So it's much more complicated, obviously, than just the simple instructions. Let me give you another example, though, that's even more stunning, and this just was in Nature 2 weeks ago. So given the ability to sequence whole genomes suddenly, even though it's still expensive, it's not as expensive, this group took 3 sets of twins, identical twins, one who had multiple sclerosis, one who did not. On one set, they sequenced the whole genome. And not only sequenced the whole genome, they sequenced analyzed the whole epigenome, all the epigenetic changes. They looked at the different setup in their T cells to see what, because it's an autoimmune disease, they looked to see what differences there might be in T cells. How many differences did they find between these two people, one with MS, one without? Zip. So those of us in the genomics business kind of took a deep breath because even the notion that there might be rare things in there kind of shocked us. So there may still be something we just didn't find, but they looked at 2 other sets doing whole genome association studies and didn't find any differences either. So there's obviously more work to do, but it's fairly clear that genetics is not necessarily destiny. So Steve will like this slide. So the battle is genomics suddenly just kind of worthless? As I said, it's very sexy. I don't know if you can read that. It says, "Who cares? The emperor is buff." So there's this hot debate whether genomics will actually contribute anything to medicine, whether it'll really provide anything that's significantly clinically actionable, or whether it's only going to be useful in just a few occasional cases, like I've mentioned in cancer and some pharmacogenetics. I think the fact that there's been a lot of early over-the-top statements about how this was going to transform the world by people like Francis Collins, which was aimed not so much at the general public, but at the funding sources, I don't think it's helped this perception, and indeed may have provoked the notion further that it's been overpromised and it's not going to deliver. I tend to think, though, the people that are on both sides are wrong. The ones that believe that genomics is going to be utterly transformative of medicine as in genomics, qua genomics, and those that think it's not going to contribute everything or anything, and who promote it as kind of a wasted start at personalized medicine. So genomics is ultimately critically important, but maybe not so much as a clinical entity, at least not for now. So I want to talk about this notion of pathways and come back to the black box and where genomics is actually leading us. So for those who don't have to look at these things regularly, this is kind of a very simplified view of 4 of the major pathways that we study. This is TGF-beta, insulin, hedgehog. Most of these are named after fruit fly mutations. It's a long story. Wnt. And in essence, what all a pathway is it has some kind of receptor on the cell surface, and some kind of stimuli will move that receptor. And it's like a relay system that happens with a bunch of proteins that then interact, that go to the nucleus and do something with the DNA and get the cell to respond. So if you think about each of those elements of that pathway, each of those little proteins, each one is encoded by a gene, and you can imagine if there's a slight mutation or a little mutation in one of those genes encoding one of those proteins, that the response may not be correct. It may end up with a cell making too many copies of itself and being cancer. It may end up with a cell not recognizing that that was a photon of light and it results in blindness. So there's these pathways that all this genetics is collapsing on. And all of a sudden, instead of having 20,000 random genes out there that you're trying to link to diseases, in essence, what you're doing is linking these genes to each other around these pathways. And there's only a couple hundred of those. So all of a sudden, we have something that's beginning to sort out what's really going on in the black box. And a thing that I find most amazing and most interesting about this is if you look at where some of these diseases end up mapping. I'll just look at Wnt for right now. So, whoops, wrong button. Vitreoretinopathy, colon cancer, and osteoporosis, one type of it, are all mapped onto this Wnt pathway very close to each other. So think about the implications of that. These 3 kinds of patients, someone with an issue with their retina, someone with colon cancer, someone with osteoporosis, are in 3 different clinics at this moment, being approached 3 entirely different ways, under 3 different specialties. But in fact, at the level of their phenotype, when we think about molecules, they're actually much closer related than they are to other types of vitreoretinopathy or to other types of colon cancer. It's really a stunning thing, and just suggests that the whole field of medicine is going to have to be reorganized around something like pathways rather than organ systems. And this is what genetics is actually helping us with, and genomics is helping us with, is begin to sort out what are these kind of weird little categories and give us understandings of what is going on in biology. And that can't be underestimated. It's pretty exciting. It's a long ways off, and more data will help. So, but again, it's not helping us a great deal with medicine now. It's helping us to understand what the biology is and what goes wrong. So I want to come back to the other side then and think about what it is that phenotype does and how it might contribute its way back into this box. So I want to start first with the notion that even though people have brought about personalized medicine as a discipline or talked about it as a result of the tremendous advances in genomics, it's a bit of a misnomer. Medicine has always been personalized. It's always been based on phenotype, at least phenotype that we could see and measure in some way. So indulge me, I keep hitting the wrong thing. So indulge me while I describe a 4th century BCE case study. So Apollo is feeling kind of crappy, goes to Hippocrates. Hippocrates will carefully observe Apollo and listen to him. And Apollo will tell him he's feeling kind of out of sorts, kind of blue, he's very low energy, kind of listless. And Hippocrates will consider what Apollo looks like. He might take his pulse if they did that. And then he diagnoses Apollo to be suffering from melancholia, or an overabundance of black bile, one of the 4 humors that were the definitions of phenotype in that day. So he prescribes to Apollo some dietary changes, abstinence, and a bowel purge. And so Apollo walks off with his prescription, gets his bowel purge at whatever the CVS was in Greece back then, comes back to Hippocrates in a week and reports he's feeling a lot better, probably because he's off of that regimen, but not quite back to normal. So Hippocrates looks at him again, weighs the humors, feels his temple, and says, okay, well, so the choleric humor is a little out of whack, but it's getting much better. So he sends him with another regime, this time eating some kind of weird vegetables. And Apollo comes back in a week and says, great, everything's fine, I feel incredible, thanks Hippocrates profusely, stops by the desk on the way out the door to make his next appointment and pays his 2-goat copay. So what did Hippocrates do? He carefully observed the phenotype of the patient in front of him, the phenotype he could see. He mapped it onto his state of medical knowledge and his knowledge of biology, and he prescribed the right medical treatment at the time for the phenotype he saw. So the right treatment at the right time for the right patient. It sounds like Hippocrates was pretty much a personalized medicine physician. So there's a couple of things we should think about when we start talking about personalized medicine and science. First is that phenotype is more important than genotype. Phenotype is immediate. It's not about potential. It's not about risk. It's actionable, even though the action may be wrong, depending on knowledge level, and it's trackable over time, stuff that everybody's talked about. So in essence, what we've been doing over all these years of advances is simply seeing deeper and deeper and deeper into what phenotype is. We've gone from kind of full person to organ systems to cellular levels and now into molecular levels. And that probably should be proteins instead of DNA out there. But I couldn't redraw this thing. I couldn't figure out how to do that. But basically, we're just beginning to see deeper and deeper, but we're still assessing phenotype. And we're getting phenotype closer and closer to the biological complexity we're interested in. How do we know if these kind of molecular measurements of phenotype, though, are really correlated with the diseases we're interested in, or the diseases we're studying? Too often, still, we don't. You only have to think about prostate-specific antigen, or PSA, which can indicate prostate cancer, or can lead to a lot of unnecessary surgery and some pretty brutal sequelae, including seeing Bob Dole doing Viagra commercials. So these telltale markers, though, and these proteins, as people have mentioned, there's thousands of them in the literature. Everybody publishes, we found this protein associated with this disease. But so few of them ever turn out to be valid and truly not just associated with the disease, but absolutely indicative of the disease. And this is yet another graph showing how few there have been that have been approved by the FDA. Not that the FDA is the final word; it is the final word in some ways, but have been shown to be medically useful. So the challenge is really, how do you validate these things? And there's lots of attempts to try to do that. It's pretty tough. The proteins are messy. They're unlike DNA. There's thousands of them in weird combinations at different levels and different concentrations throughout blood and other tissues. It's been practically impossible to sort this all out. So from my perspective, coming here from the genomics world, recognizing the limitations of what it is we're trying to do is incredibly exciting to me to hear these talks this morning. There hasn't been any real transformational leap in proteomics since the immunoassay 50 years ago. And it's really very exciting to hear the stuff going on at SomaLogic and a few other places that are actually going to be able to help us leap over this divide. So let me talk about one other thing. There's also a sense that if we really don't understand the biology, that there's really no reason to believe we can do good diagnosis and treatment. And I don't think that's true. I think there's a lot of things that are fairly actionable right now, and phenotype is hugely actionable if you really get a valid phenotype. But there are attempts to start to pull the black box together. So the simpler pathways I show you, this is more like a standard kind of pathway. This is what's getting into what's called systems biology, or what we used to call, in my day, physiology. The idea is, can you figure out what all the parts are? Can you figure out what all the inputs are, not just genetic and epigenetic, but environmental? Can you tie that to outputs? And there's a couple approaches to try to do this. One is to just take the vast literature and aggregate it together and try to pull patterns out of that. And there's whole companies dedicated to that. It's not going to be hugely successful because it depends entirely on what we find in the laboratories. So it's at the mercy of the slowness of our research. The other approach, which I think is kind of fascinating, is a company called Gene Network Sciences and a couple others who've taken advantage of computer power, and then they just throw all the raw data in there. What are all the phenotypic data? What are all the genetic data? What are all the epigenetic data, environmental data? And they let the computer figure out the model and begin to model a system and saying, if you begin to make a tweak here or you make a slight alteration here, it'll have this kind of output. And it's hugely promising, not proven yet, but I think it's only going to be those kind of approaches that are ultimately going to fill this in. But the good news is we don't need to get there yet. Someday we will, but we don't need to get there yet to make a really, truly personalized medicine. So this is the moment, I think, for phenotype. We have an amazing opportunity to realize some significant advances now. Despite our fascination or even our obsession with genomics and genetics, I think advances in, this is almost heresy, but I think advances in proteomics will have the most immediate payoff in terms of detecting and tracking and treating and maybe even preventing disease. It's not antagonistic to genomics. Indeed, I think proteomics and genomics actually have a great deal to learn or build from each other around what's in the middle there. But in terms of immediate clinical impact, I think the transformative advances we're starting to see are going to let us see deeply into phenotype for really the first time and in real time, not as a measure of potential risk, and these advances can't be understated. So the challenge, of course, is to have a receptive healthcare system that's able to take in these new things and get them to patients. So I look forward to hearing more about those challenges and opportunities the rest of this afternoon and tomorrow. So thank you very much.
Are there any questions?
Oh, God, okay, yeah.
You're the last of the genomics speakers, I think. This was really a question for Richard, but maybe somebody else will comment on this. We talked all morning long about, oh, thank you. We talked all morning long about genetics and genetic diseases as if every mutation associated with the disease was a germline mutation. And when we talk about complex diseases like cancer, we talk about somatic mutations. So if you take a buccal swipe and you do a DNA analysis on that, that will have nothing to do with the mutation that has taken place in your pancreas, for instance, and led to pancreatic cancer. And nobody has talked about that yet. So I hope before we get off the genetics, either the power or the criticism of genetic analysis associated with diseases, somebody would talk about that.
Yeah, I think it's actually even a little worse than that. If you take section of liver, normal liver, and a section of normal liver a little further down, or even a section of normal liver and a section of fat tissue, and you do the genome sequences, they're not the same, even though they're derived presumably out of the same cells. So there's lots of stuff that goes on in the course of development. It's pretty
Larry did a calculation of this. Larry, why don't you say what you found?
What I would say is that there's supposedly 10 to the 14 replications of cells from the fertilized egg to the human, adult human. And every one of those cell divisions is associated with mutations. And unless it happened at the first division, it becomes a minor subset of the sequence. And so when you sequence any particular thing, you get more or less the starting genotype. So you're missing all the somatic mutations unless they happen very early.
Right. And this is a significant challenge when you're comparing so-called normal tissue to a cancer sample, because you really have no idea if that normal tissue was reflective of whatever was supposed to be normal in that cancer. Yeah. Yeah.
Okay.
Sorry, have people actually looked? Have people actually looked at 4 pieces of human liver from the same person, or actually done those experiments?
They've done like associations there. They haven't, and they've looked at some. There's been some exome sequencing. Not entire genome, because it's still kind of expensive and people don't want to know the answer. Once it gets cheaper, yeah, they'll do that. But there is enough material out there to show that there's changes as you go across tissue types, even within tissue.
Like single nucleotides, but it'd be, I mean, that's the central dogma, right? Every cell is the same. I'm just wondering if it'd be interesting for somebody to check.
Yeah, no, it's not. It's really not true that every cell is the same. In fact, every cell tends to have differences. But I know what you're saying. The notion was that once you got mom and dad together, a little more than 50% for mom and 48% from dad or whatever it was, because X and Y chromosome have different things. But it would be the same all the way through, but it doesn't because of the changes.
Larry had
So most of what you said I thought was just wonderful and we could all agree with, but there's one thing you said that made me a little nervous.
Okay.
Which was that there's only 100 or a couple hundred pathways. And that seems to me an odd statement because, of course, all of the reactions are connected, at the very
Right.
least through ATP and the like, and what we define as a pathway is sort of for our convenience and understanding rather than actually divided up in the living organism. So why do you think understanding pathways is going to be easier than understanding genes, and why do you think there's so few of them?
Okay, that's actually two good questions. So we don't know how many there are. There's like 200 and some in BioCarta right now, I think. So, but the connectivity between them adds new layers of complexity. But what's interesting is that there seem to be critical nodes that these pathways will, when they interact, will share, and those are targetable. Those are the kind of things we want to start looking at, not the kind of off pieces, but how is this thing organized? And that's a little more tractable a problem than just trying to figure out from the beginning how a gene does its thing. I mean, RNA knockdowns and things like that we can do, but if we can begin to build a biological chart or a biocarta, then there's a way to think about organizing how you do intervention type stuff. But it's hugely complicated. As you know, these are very simplified versions of this. So point well taken.
any other questions?
This may be naive, but whatever became of the expression junk DNA? Is that still an applicable term, or has that gone by the wayside?
Yeah, there were a couple numbers thrown out this morning. About 5% of the human genome is highly conserved, and we know that by looking across other mammalian genomes, just figuring out what did evolution think was important, and comparing other humans. In that 5%, about a third of it is protein coding stuff. The rest we don't know what the hell it does. Some of it's some interesting RNAs, some of it's other stuff. And then in the genome-wide association studies, we begin to find strong associations in areas where there don't seem to be anything conserved and have no idea what's going on there. So junk DNA is quietly disappearing as a term because it's basically DNA that we don't know what it does yet. So.
Well, but you know that, for instance, again, for oncogenes, when Genentech first worked on the RAS mutation, they first published that you needed an exonic mutation for this to be oncogenic. And then when they produced that mutation, it turned out that it wasn't by itself oncogenic. There was a second mutation in what was then called junk DNA. It was an intron. That mutation stabilized the pre-message, and so there was more of the mutant protein made. And in order for this to be oncogenic, you needed two mutations, one in the coding sequence and one in the so-called junk. And that affected the stability of the message, the amount of protein made. And in order for this to be an oncogenic event, you needed both a missense mutation in the coding part of the gene, and you needed more of that mutated protein so that without that, you wouldn't have an oncogenic event.
Yeah.
That was in so-called junk. The second mutation was in so-called junk DNA.
No, I was just, you know, evolution's had like millions of years to try to tinker with all this stuff, and we're trying to catch up.
And I promised you one last question.
Yeah, when you were speaking about the different phenotypes or different body systems that you have a colon abnormality and a brain abnormality, and you have these different things, and you have different subspecialists seeing you for different things. It made me think about what I do in ultrasound from when babies are very little, and how you can see some, you can make these associations because you're seeing through the whole baby, and you can see very quickly what all the associations are. You might have genetics people mostly that you're working with, rather than the subspecialists beyond that, although sometimes they're involved. But what it made me think of is, geez, what if early on, at 10 weeks or 12 weeks, you could see something in one organ system and diagnose it and do genetic therapy somehow in the baby early in development that could reverse the problems. So
I follow you up through the be able to see something going awry early,
Right,
and be able to,
things, you know,
and then the notion would be to be able to fix it somehow. I don't think gene therapy, though, is going to be an answer, but there may be a small molecule kind of thing. Or
I mean, because the fetus is in an environment, you know, and epigenetics factors in a lot when you're talking about very rapidly developing cells in an embryo that could be, you know, tweaked.
Or the mom could just put down the martini.
But I mean, maybe it's a couple hundred years away.
No, it's a fair question.
Thank
Sorry.
you, Fintan. Great talk.